I'm going to touch on a number of themes that have come up multiple times over the course of the day. And I feel very much like I'm preaching to the choir here. But this is coming from a perspective of having spent a long time in the area of cognitive neuroscience, particularly the area of functional MRI and functional neuroimaging of cognition. And there's a history as to how these methods got kind of integrated into work on cognitive aging. And because of that history, it's fair to say, and I think Natasha and Audrey would agree with me, if there's an area of aging research in which issues of diversity, sample composition, and just reflection on the implications of sample bias and selection bias have been underappreciated, it is that area. So that's the area that I'm kind of coming from here. Coming from other sorts of traditions in the field of aging research, and particularly Alzheimer's research, some of these issues have been around forever. Anyway, so before we start, I should just acknowledge the little bit of research that I'm going to describe from my own lab was supported by the National Institute of Aging, who support pretty well all of the research in my lab. So thank you. Does anyone here? Thank you, and give me a good score on my next round. And also, sadly, no competing interests, as has been said previously by others. So I am going to start by just quoting the what is still, I think, the current statement of the National Institutes of Health about diversity requirements. So it is the policy of the NIH that women and members of minority groups and their subpopulations be included in samples. And the research plan should describe the composition of the proposed study population in terms of sex/gender and racial and ethnic groups, and it should provide a rationale for the selection. Everyone, in a sense, pays some kind of attention to that, because if you don't, you won't get your research funded. And this is a pretty good example of how people do that. This is actually, for those who fill in NIH grant applications, this will be very familiar to you. It's an inclusion enrollment report in which we are mandated to say how we plan to populate each of these boxes. The boxes are separated by sex, they're separated by racial categories, and they're populated by ethnic categories. And basically what most people do, and I include myself in this, I'm not being hypocritical here, we have been as bad at this as anyone else, is that we have as our recruitment goals trying to produce a set of numbers in these boxes that more or less roughly corresponds to the demographic representation of those groups in the population from which we are sampling. So, for example, in the greater Dallas area, I will explain what the demographics are of that area, and I pretty well just take out a calculator and work out what that will mean in terms of an ideal sample composition based on that area. So you end up with things like, I think we say that I can't quite read this because it's so small on the screen, but we say that we will have 2 male and 2 female people of American Indian Aleutian descent. You know, we never do, because we can never find them and they never come forward. But the point is that that's what we propose to do. And the idea, and everyone does this, is try to produce a diverse sample along these lines, and then essentially analyze that sample as a homogeneous sample. And I'm going to argue that that is not, I think, the right way to do this. Thinking about this quite a lot, and as someone who still does this, I think it's wrong. I think if we really think that people of, let's say, American Indian descent may have important and distinctive features in the data that we're collecting that mean that we can't generalize from another group to that group, we don't do anything to understand that by putting 2 people into our sample. In other words, we should be thinking hard prospectively about what we think are the most important sources of variability in a sample, and then enriching the sample so that we can actually do justice from a statistical point of view to whether those data differ from those of another group, and actually do meaningful statistical comparisons. At the moment, with the NIH right now, the only mandated contrast that we're required to do is for sex. You have to write into your grant a section about sex as a biological variable. You basically have to say, we know sex is important and we will do preliminary analysis to see whether there are any sex differences. However, you're not even required, unless the grant has as one of its primary aims the explanation of sex differences, to power the grant to detect sex differences. They can simply be considered to be an exploratory analysis that's done ahead of the main analysis. Thinking about this, over the last few years, and particularly in thinking about it in connection with this meeting, I think that is not an optimal approach. And in a sense, it's to some degree doing a disservice to the notion of diversity. It's actually saying, well, let's just, put in a few, let's just lump people. And we have a representative and a diverse sample, therefore, whatever we find out is going to generalize. And this comes to your point about generalizing. I think it's the opposite. It's anti-generalization. So that is my kind of take-home message. I'm hoping now that there is no one from the NIH in the audience. But anyway, I think that's the point. What I'd like to do is to give a couple of very simple examples. And these are deliberately simple, and they're deliberately ones where no one is going to be surprised by the results. Just to make the point about how taking seriously the importance of differential groupings of samples can make differences to the results. So if we go along and do what we typically do, we end up with a sample. This is what one might call a diverse sample. It's got some kind of variability in it. I've made it conveniently conform to a Gaussian distribution. This can be anything. It could be brain cortical thickness, cortical volume, some measure of activation in some region of the brain. But basically, we know that if we have got what we would call a diverse sample, by definition, there must be a whole bunch of subsamples, each of which have their own variance, their own distribution. And they are, if we assume that these are randomly distributed Gaussian variables, when added up, they will give us our big parent Gaussian distribution. And what I'm trying to argue is that, having one of these subsamples populated by a very small number of people is pretty much a waste of time. It tells us nothing about those people. And all it does is add potentially error variance and also give us an unrepresentative picture of what's going on. So that is the basic issue, and I want to give just two very simple examples of that. So the point being that subsamples, most obviously, and this has been discussed many times with very good examples during the day, they can differ in theoretically and practically important ways in respect of the variable of interest. We've had some excellent examples of that in the field of sex and gender. But of course, there are many other ways that we can slice up a sample, and the same thing will be true. I want to point out, though, and give an example. The example I'm going to give here, by the way, is not from my own work, but from a colleague's work in the field of socioeconomic status. The second way, though, that not recognizing subsamples can be important is that by not controlling for these subsamples or stratifying, not stratifying is the wrong word, but segregating, if you like, the data by a particular subsample, you may actually obscure what is actually a general relationship. In other words, something that is general across more than one subsample may turn out to be actually not undetectable. In other words, the subsample label can act as a suppressor variable in your statistical analysis. And the example I want to give there is sex. Not because we haven't talked about sex already, it's because it's the only example I've got in my own data. And it's also, in this case, quite a nice one. So these are the examples I want to give very quickly. This is the example from socioeconomic status. This socioeconomic status really is basically defined in a variety of ways. In my this is from Gargan Weeks' lab, a colleague of mine in the Center for Vital Longevity. They investigated a measure of brain segregation in resting state fMRI, and they also just simply looked at mean cortical thickness as a function of SAS, where SAS was a combination of occupation and income. And you can see in these two middle-aged cohorts here, interestingly, not in the younger cohort or the older cohort. This is cross-sectional data, by the way. Nothing to be said about aging here. But you can see in the two middle-aged cohorts, pretty dramatic separation with a lower segregation and lower cortical thickness in adults who was sampled from the low SES group. Most of us, myself included, do not enrich our samples in the way that Gargan is doing right now. This is historical data that is doing right now. We can't even address this question. But clearly, it's clearly very important, and it has important implications for how we think about age differences in these functionally important variables. Secondly, this is from my own lab. This is describing simply an older cohort from a much bigger study, which is published down here. We had around about 150 older adults, roughly evenly split between males and females. And in these individuals, we had a very large number of cognitive measures. I won't bore you with what all of them are, but they were sufficient to allow us to use a principal components analysis approach to pull out a factor which corresponds to general intelligence, what we call general cognitive ability. I might just say, in recognition of what you mentioned, if we look at the verbal memory scores, if we look at verbal memory, women massively outperform men. But in actual fact, when we look at general intelligence, that is the first principal factor from all of these cognitive scores, males and females, males are slightly worse than females. The negative thing here is because of z-scoring across the whole sample. But there's no significant difference in general cognitive ability between males and females. Of interest to us right now is among the many is an exaggeration, among the several structural brain measures that we investigated here was a whole brain grey matter volume. You'll see that women have markedly smaller brains than men do. This is well known, and much smaller grey matter volume, despite the fact that they're just smart. So much for the brain size itself being a predictor of cognition. Critically, though, if we look at the relationship between brain volume, and cognition, and we simply lump all of our participants together without regard to sex, there is no significant relationship. In other words, if we say, well, we want to be unbiased with respect to sex, so the best thing to do is simply take all of our data and do a big analysis on it, we see nothing. And so that is what we see as the scatter plot. If we include sex as a variable in our regression equation, and by the way, importantly, the interaction term, so that we can know whether there's a moderating effect of sex on the effects, we see a robust relationship now between brain volume and cognitive ability. In other words, and this is sex invariant, that is, it's not that one sex is driving the effect, it's that we don't see the result, that we don't put sex into the model, but the relationship itself is common to everyone in the sample. So it's a suppressor variable. So basically, just very quickly to conclude, splitting is better than lumping. But this requires a prospective approach to sampling. And this is something that we have not done. It's something that plenty of people in this room have done. But it's something that in my field has yet to be seriously taken on board. We're going to do that. We're going to end up with large overall sample sizes to ensure adequate statistical power, much larger than the ones that we typically use. And the critical question, and this comes back to your point, is how do we decide if we're going to go down this strategy? What are the number and the nature of those subsamples? Sex is a relatively easy one. SES is a relatively easy one. Things get much more difficult, I think, when we start to think about other ways that people can differ in terms of genetic backgrounds, cultural backgrounds, etc. But we have to make some choices there. And how we make those choices, I think, and how the field makes those choices is going to be very important as a question going forward. So that's it. Thank you very much for your attention.